right? Just 1 minute or 60 seconds. That's all. Rinse it with water. Then we're gonna add our iodine for 60 seconds also. Rinse it with water. Notice that everything right now is purple, right? We have these bacterial cells that are rod-shaped. Rods look like Tylenol capsules, right? And then your little circular shapes look like spears, peas, whatever you want to call them, but they're circular shape. They're cocci. So right now, the cocci and the rods are both purple, right? So we're going to add the colorizer, which is the shortest step, 5 to 7 seconds, right? And once we added the colorizer and rinse it off with the water, this is what we're going to see. The rod-shaped bacteria, bacteria are now clear. The circular bacteria are going to be purple, right? So the colorless bacteria are that way because they're going to be gram-negative. Remember, gram-negative bacteria lose their pigment during the colorizer stage because it causes those porins to pour out the solution. Everything just leaks on out. All the stain leaks out. And then you add your safenin for 60 seconds. Rinse that with water, and that's your final product. Right? The mixture of gram-positive and gram-negative bacteria, gram-positive being purple, gram-negative being pink. All right? So this is what it looks like when you look at it underneath the microscope. So underneath the microscope, that specimen would be the pink rods there, those longer structures, and like these purple would be your purples. That's actually Staphylococcus, the purple, because staph is always going to be clustered together like grapes. So pink and purple tells us that there's gram-positive and gram-negative bacteria in that sample. All right, so gram stains are very, very important. We use that as one of the first lines of treatment, right? Like I remember when I was a kid, they swab my throat, and for like they thought I had strep, they would swab my throat, and they would bring it off to the lab, and the lab would put it on the plate, and then they would make that drug, and then they would stain it, and then all these different steps. Right nowadays, you just go to the doctor. They swab that bad boy right there in that rapid test, and the results are like 15 minutes. I'm like, dang, I love this. I love this. But nonetheless, though, this process is a fact check, though. All right? So even though we do like the rapid test, in some cases they do fact checks to ensure that this is in fact what it's supposed to be, especially in the hospital settings.
Again, how long was the safranin?
Safranin is 60 seconds.
Okay.
So safranin, safranin. The only step that's different is that alcohol step. It has to be the shortest one, right? Because alcohol will wash it smooth out the slide.
Yeah.
Yep. Great question. So, when talking about the gram stains, you're also able to use it to detect bacteria in clinical samples, right? Clinical specimens. So you can have a specimen and you want to see whether or not there's bacterial growth in that. You can do so by doing a gram stain. Also, for unknown, so in my last institution, I gave my students unknown bacteria to identify. It was a series of steps, and we do a little biochemical testing here too. So you'll see that process of identifying an unknown bacteria, which is so easy. Once I teach you the steps, it'll be like clockwork. In all my classes, whenever I teach my students the process, it comes easy because I showed you everything. You just have to do it by yourself now. That's it. All right. So last thing, gram stains are mostly used on or done on young, actively growing bacteria because if you have an older strain of bacteria, it may not stain properly. It can give you some false results. So that's why in the lab you get fresh samples. Right, you get fresh broths, you get fresh plates, right? So that to ensure that everything is fresh and actively growing. So we're gonna also talk about the acid fast stain. So acid fast is interesting because it binds only to bacteria that have this waxy material present in its cell walls. All right, they have this waxy material present in its cell walls. And so thinking about wax is a lipid, right? So lipids we know are hydrophobic, they're not gonna readily interact with water. So the staining have to be very specific for this process because it won't retain the other stains as they should for a gram stain protocol. So that's why we're going to be using acid alcohol for this particular protocol because this waxy material is not decolorized by acid alcohol, but regular bacteria are. All right, regular bacteria can be decolorized, and that's what we want to see. So what are the bacteria primarily known for being acid fast positive? Mycobacterium and Nocardia. So Mycobacterium tuberculosis, TB, is where we would use this as a diagnosis for. So if we see someone come in presenting symptoms that look like TB, then that'll be probably one of the first tests that you're gonna run to see whether or not they have Mycobacterium. All right? So let's see what that protocol looks like. So for acid fast bacteria or non-acid fast bacteria, we're gonna do the same steps. We're going to add the primary stain, carbon fusion. I won't make you know the time because I sometimes get it wrong. I know it's like 5 to 7 minutes, but like I don't make you know the time for that. But nonetheless, though, carbon fusion, you add that as the primary stain. And that means that everything right now will be red. Carbon fusion is red. So everything right now will be red because that's the reagent that we added, right? And so after we add the carbon fusion, we'll rinse that with water, and then we're gonna add that acid alcohol still very fast because alcohol will wash that sample, clean off the side. And so acid fast bacteria will be red. So remember I just said that non-acid fast bacteria, they're not non-acid fast because, they are non-acid fast because they do not have a waxy material in their membrane. So since they do not have that waxy material, then the decolorizer will be able to work. And it's gonna decolorize those non-acid fast bacteria. So that's why you're gonna add your counter stain, your last stain, your methylene blue, and your non-acid fast bacteria will be blue. And so the red will be the color because they're gonna still be red. So mycobacterium will be red, nocardia will be red, but if I were to do staph will be blue. Staph is non-acid fast, right? E. coli is gonna be blue because E. coli is non-acid fast. Get that? What differentiate between those different types of bacteria?
So is the waxy material within the peptidoglycan cell wall?
So within these bacterial cells, yes, you have that material present. So the acid is actually called mycolic acid. I just didn't say it, but like that's the name of it.
And it's just mixed in, it's not like an extra coating outside
It's not
it?
absolutely. So it's not an extra coating. It's just within that membrane. Yep, it's there. They're not allowing that stain to be contained there. All right. Get a packet. Are you? Good morning. Thank you very much, Taylor. All right, so looking at a micrograph, so I don't think I said this before, but micrographs is like a picture, right? It's basically a picture taken with a micrograph. So this micrograph here shows what this would look like for a light microscope, looking at Mycobacterium tuberculosis, the red that you see there, because it's an acid-fast bacteria. All right, so it's an acid-fast bacteria. Now, the next stains that we're going to talk about are special stains. We don't necessarily do some of these. Some of them we do, though. One of them we're going to do. But for some of them, we just look at it. We use it as a reference point. And so these are important for distinguishing different parts of microorganisms, and the names kind of tell you what the structures are. So the capsule stain will highlight the capsule structure. The endospore stain will highlight or emphasize the endospore. The flagella stain will highlight or emphasize the flagella or flagellum, depending on how many that the microorganism has. So, when looking at the capsule stain, so capsule stains are negative staining. So if you remember, I mentioned negative staining is when you stain the background. Remember that? I was like, negative staining is done with acidic dyes because acidic dyes are going to be the negatively charged dyes, and they're negatively charged. Our cell wall is negatively charged, right? So negatives and negatives are going to repel, right? So that's why the stain won't be retained in the cell wall. It's going to just stain the background, right? So this is what happens here. So capsules are a gelatinous covering that do not accept most dyes. And so capsules are going to be present on some bacteria. I'll tell you guys when those are. But for the purposes of what we're talking about right now, they're using India ink or nigrosin, which are acidic dyes that we mentioned last time. We mentioned nigrosin last time as an acidic dye. And so these dyes are going to contrast with the background, right? Contrast the background with the capsule. And so the cells themselves are being stained. So the capsule appears as a halo around the stained bacterial cell. Like you're going to see it. It's big as day. If the halo is there, that means there is a capsule. If the halo is not there, there is no capsule, right? So if we look at this example right here, we see that we have these rod-shaped bacteria with all these halos, right? So you see those halos, those bright white outlines around those? The background's dark, but that halo is like, bam, bright, visible, I see it. So these have capsules present, all right? Make sense?
Yes.
All right, so we don't really do capsule stains in labs. I'm not really a fan of them. They're cool though, but like, so this is what I am a fan of though, endospore stains. So endospores, I've mentioned them to you so many times, but endospores are really interesting because they're resistant to most environmental conditions, especially extreme environments, right? They can sit there and thrive in dry environments. They can thrive in extremely hot environments. They can thrive in cold environments. Just depends on what we're talking about. But nonetheless though, they are dormant structures, meaning that they hang tight until activated by some type of stimulus, right? So they can hang tight. So dormant structures inside some cells that cannot be stained by ordinary methods. So we have to have a special protocol for endospore staining. So we normally use the Schaefer-Fulton method because the Schaefer-Fulton method is quite intense. because you're gonna be using your primary stain, malachite green, which is gonna be green. Yep. So malachite green is green. And what we're gonna do is we'll have our slide on the rack. We're gonna make our smear, right? We're gonna have that water, the bacteria, edge dry it, heat fix it. Bam, smear's done. So we're gonna add malachite green, and it's gonna sit on there for 10 minutes. So once you add the malachite green, it's gonna hang on there for 10 minutes, but you're gonna pick up the Bunsen burner, and you're going to apply, or have that, like literally pick it up and hold it to where the flame runs across that slide for the whole 10 minutes. You're just gonna sit there. Just 10 minutes, I kid you not. And it's cool though. So why are we doing that? Well, the reason for that is because when you see a bacterial cell, a gram-negative bacterial cell that has this endospore, it's going to be very difficult for that stain, any stain, to penetrate that endospore coat. So that is the cell. That's the rod-shaped cell. And then this is the endospore, right? That endospore coat is very, very thick, right? So the coat itself is like really thick, very, very, very thick. So the spore coat, so thick that you have to apply that heat in order for the reagent, the malachite green, to penetrate that spore coat. So once the heat is applied and that spore coat is penetrated, you now have that spore coat appearing as green. All right, because of the malachite green. And so once you add your primary stain, your malachite green, you're gonna rinse it with water. After those 10 minutes pass, then you're gonna colorize those cells with water. It's very simple because, you know, the spore coat not gonna penetrate it anyways. So you might as well just rinse it with water. And the cells are kind of vulnerable right now. They're vulnerable because you just had heat on top of them, right? So just rinsing them with water is sufficient. And then you'll be able to hit them with the counter stain of saffron. Saffron is what color? Remember that color? Saffron is red, reddish pink, right? So saffron is the reddish pink color. So that's why when you do an endospore stain, if the cells have endospores present, the cells will be pinkish red and the endospore will be green. If there are no endospores, then the cells will just be red. There will be no green, all right? Because there's no endospores stain. This is what it looks like. Looks pretty cool. So looking at it, you see like these little rod-shaped bacteria with those little green. All right. So now the flagella stain is one that we normally don't do. We just look at it. So like flagella, we know are used for movement, locomotion. And so these are very, very narrow, very slender structures, right? They're long too, but they're very slender. So it's kind of hard to see them. So there needs to be an extra coating on those for us to see through the light microscope. So we add a mordant to it as well as carbofusion to make it thicker, right? So we add a mordant. So the mordant we know is what's going to help coat the structure, right? And make it appear larger. And so this makes it this one even like microscope. Also, why do we do the flagella stain? The reason why we do it is to see whether or not there are flagella present, and then how many are they, and then where are they, how are they arranged. So we're gonna talk about that because flagella can be they could be on like just one end, a pole, or they could be on both poles. They can be all over, or you can have like a tuft of them in one area. It just depends on what we're talking about. All right, so that's why we do the flagella stain, to see the flagellum arrangement, or flagella arrangement, or flagellum. Singular versus plural, that's why I was doing that. Okay, so here we see the flagella staining protocol showing us what this looks like. So we have this little fella right there with the flagella. We have a tuft of flagella hanging out over there. Tuft means a bunch, I'm sorry. So tuft, T-U-F-T, means bunch. So you also have like some like all over it. So you see why we do that? To see where those flagella hang out at on the structure. And that's very specific too. Like some bacteria are known to have them in one spot or to have them all over. It's very specific. We know that based upon our work. And we're done. So question for you guys. Which of the following is not, and we don't shout it, right? I had to think like, wait a second. We don't shout it. We're gonna read it. We're gonna wait a couple seconds and then we're gonna answer it. So which of the following is not a functionally analogous pair of stains? Is it A, nigrosin and malachite green? Is it B, crystal violet and carbofusion? Is it C, saffron and methylene blue? Is it D, ethanol, acetone and acid alcohol? Or E, all of the above pairs are functionally analogous? So do you know what functional analysis means? You need a little help on that? Some of you guys do, some of you, because I was sitting there, I was like, do you know what that like means in this context? So, in this context, we're meaning functionally analogous, saying like, in its protocol, in the order in which it is in this protocol, are they similar? Does that make sense? So like, you have these reagents in different protocols, right? Because I know you know that for instance, nigrosin and malachite green is not in the same protocol, right? So in its separate protocol, is it similar in? Yeah, you get that? Yeah. It's like functionally analogous, which is not, yes.
So
Right.
then you wanna know the homologous, functional homologous?
No, it's just just which one of these, because some of these are going to be analogous to one another, but then which the one is not gonna be, yeah. I see why you asked that though, because it's the opposite of that, but it's think about in the context is, yeah, this is not the two negative ones, but I understand why you said that, I really do, I can get that. And so I'm letting you guys turn the pages because I just want to give you that set. All right. So we walk it back. All right. So let's talk about it. Who put on 3? Show me your answers. A, B, C, D, or you can tell me. All right, argument 1, 2, 3, 4 works too. If you like, I don't do sign language, whatever. All right, and that's E. So 1, 2, and 3.
B.
I see B. You got
B.
D, B, D, B,
No,
A. Huh?
A.
A. I'm sorry. A, B, E, C. A, you must have seen the whole thing. All right. So with that, the correct answer is actually who said B? All right, so when looking at this particular example, so you have the different protocols, and these are not gonna, you know what though? I just thought about that. What protocol are we talking about? Carbofusion is in more than one protocol.
Yeah, and so it's a primary and.
It's a primary in one, but it's a counterstain in another. I just thought about that. That's a trick question. I don't like that question. That's why you picked the A. Yeah, that makes sense. It just hit me as by myself thinking about it. All right, so ideally though, we're talking about the protocol in which crystal violet is the primary stain and carbofusion is the secondary stain, the counter stain. So if it's functionally analogous, it would have been crystal violet and say malachite green, right? Because crystal violet is the first stain and malachite green is the first stain in its separate protocol for gram staining and acid fast staining respectively. No, not acid fast, endospore staining respectively. Does that make sense? Yeah, I don't like that question. Look at the face on that. All right, so the next question. All right, which of the following pairs is mismatched? So I can use a capsule stain. I can use a negative stain to see the capsule. That's A. For B, I can use a simple stain to see the cell arrangement. For C, I can use a negative stain to see cell size. For D, I can use the gram stain for bacterial identification. Or E, none of the above. Okay, so think about it for a sec. Oh, snap. Make it touch the screen. It won't let me go back. I saw it. Okay. So, which answer were you gonna go with?
C.
So why were you gonna were you really like, okay, so for this one, the correct answer is C, but did anybody like really was gonna go with that one? You really was? Why? Oh,
Because
no. You don't really, say, you don't really look for the size. And then we have it twice too, like, it's like, yeah. You wanna look at that, cause you have it twice. But yeah, so we don't really look at the size for this negative stain, we only look at the capital. Well, not only, but the negative staining is for the background, right? And in this instance, we talked about it for the capital staining. Size, no, it's just to make it to focus on the background and show the structure that we're gonna have type of, Alright, so yeah, with that being said, chapter 3 is done. Alright, good deal. So we're gonna come off of that and jump into chapter 4. Okay, so did you guys look over chapter 4? What do you think about it?
More in depth of chapter 1, more explaining.
Yeah, so thank you very much for saying that, Regina. Regina said it's more in depth of chapter 1. So essentially, we're gonna see that for all of our chapters. They're gonna go in more in depth from chapter 1, right? So chapter 1 gave us like a nice little welcome to the course. This is pretty cool, right? This is great information. Let's talk about this in more details in specific chapters now, right? Okay, so when looking into chapter 4, we're gonna focus on the functional behaviors of prokaryotic and eukaryotic cells. Right? And so when looking at that, that means the first, the first, maybe 80 slides covers the prokaryotes, and the last few slides hit the eukaryotes. So we're going to take a lot of notes today, okay? Chapter 4 is intense, and chapter 4, I don't know if you looked at your study guide, but you have a lot of questions on chapter 4. And it's a lot of specific questions because I can't be broad with you in chapter 4, right? Chapter 1 is jumping straight into it. Here we see the bacterium Serratia. So Serratia is here on some tissues, and Serratia's, this rod-shaped bacterium, is really interesting in that it has abilities to adhere to surfaces. This particular bacterium causes some of those gastrointestinal issues. It even is one that could be considered a nosocomial infection, right? What's nosocomial infections? Hospital-acquired infections, right? And that's because Serratia can be underneath our fingernails. We can accidentally carry that place. That's why it's so important with the hand washing that we did, because this is one of those that will, I hate to say will, but could be present within the hand washing if you don't do it correctly. All right? You don't want to give anybody that. So, when talking about prokaryotic and eukaryotic cells, this is going to be a review, right? Because we talked about these structures before. Just to go into a little bit more detail. And one thing that I like to do, I know this is going to seem really remedial, but I do this for myself when it's something that I'm trying to learn on my, because we're always learning stuff, right? We don't know everything. We're always learning. And so I think for me, one thing that I use are drawings, diagrams, concept maps, things of that nature. So for me, what I would do is make myself, going back to school, a Venn diagram where I would say, okay, we have prokaryotes over here, and then I have eukaryotes over there, and I'm able to differentiate between them, what do they have alike, right? So they both have these structures here, right? So I would do that for myself. I know that probably sounds like we're going back to school, K through 12, but I promise you, it helps. I used to make my students do that in my other classes, so that way they could see it for themselves, because me telling you is one thing, but actually doing is another. So this is something that will help you do it, so that way you'll have it. And then as we talk, I'll fill it out while we talk over here. All right? So when talking about prokaryotic and eukaryotic cells, what do you guys remember about those? Anything specific? One thing that you think would differentiate between the two of them? Perfect. All right, and let's see, your name, pretty name, Zinc Lalik. Yeah, pretty name. Okay, so that is absolutely what it is. So we know that pro means before, right? Karyote refers to nucleus. So this is the word that refers to cells that existed before there was even a nucleus, right? Eukaryotes, U means true, karyote means nucleus, so a true nucleus, right? And we said this in chapter 1, right? So this is a little review. We said this in chapter 1. So this is where you slap this into your Venn diagram. So in your Venn diagram, you can slap all these things in there, because when talking about prokaryotes, these are the differences right now. Prokaryotes usually have one circular chromosome that's not going to be present in a membrane, right? And that's because they do not have any organelles, right? They do not have any organelles. They do not have any phospholipid membrane enclosed organelles. That's why it's not in the membrane. All right? You also have this thing called histones. So histones are actually involved in the processing of eukaryotic organisms, well, not eukaryotic organisms, processing of DNA in its arrangement. So histones actually allow us to have our DNA like coiled up really tightly together within the cell because we have so much DNA. Prokaryotic cells don't have that, right? They don't have any histones to help with that, like organizing of our DNA, of their DNA, not ours, theirs. So also, we know that the cell walls look a little different in prokaryotes versus eukaryotes, right? We know that bacteria have peptidoglycan cell walls, right? That's the carbs and proteins, right? Whereas archaea do not have peptidoglycan. They have something similar, and it's the pseudomurine cell walls, right? So far, we're seeing some differences. Also, how do they divide? Remember I mentioned this before, binary fission, bi means two, fission, separation. So one cell will split to form two cells. Very simple process, only like a few measly steps. Really easy compared to what we see in eukaryotic cells. All right, so these are some of the differences between prokaryotes and eukaryotes. Now let's look at eukaryotes. Eukaryotes, they have paired chromosomes. We know that, right? Because humans have how many chromosomes? 46, right? So that means we have 23 pairs, right? And so when looking at that, we have our chromosomes inside of a nuclear membrane, right? Nuclear membrane. And that's going to be an organelle, a type of organelle, right? So we have organelles. Eukaryotic cells have organelles. We also have histones in eukaryotic cells because those histones help with organizing that DNA and making that being very structured within the cell. We know that, okay, so let's talk about eukaryotic cell walls. So when talking about cell walls for eukaryotes, you have animal cells, plant cells, fungal cells, and protist cells. Animal cells do not have cell walls. All right, animal cells do not have cell walls, but plant cells do, fungal cells do, some protist cells do, right? So eukaryotic cells, they have polysaccharide cell walls when the cell walls are present. Animal cells don't have it. That's why that's like that. That's why that's written like that. Animal cells do not have cell walls. All right? And then we know that eukaryotic cells divide by mitosis, right? They're prophase, metaphase, anaphase, telophase stuff. Great stuff, right? Yeah. All right, so how are they similar? Well, the one thing that makes prokaryotes and eukaryotes similar is that they both have plasma membrane. All right, they both have DNA. All right, they both have cytoplasm, and they both have ribosomes. Cause it's like I'm telling you all the differences, but like, how are they alike? So I'll say it as I wrote it. It's kind of small. Plasma membrane is the first one. DNA, the second one. The third one is cytoplasm, C-Y-T-O-P-L-A-S-M, cytoplasm. And then the last one is going to be the ribosome, R-I-B-O-S-O-M-E-S. All right. All right, so I asked this question already, but we're gonna ask it again. Why not? Okay, so on the next slide, what is the main feature? All right, so no nucleus is the main thing. All right, so the next section is actually kind of fun because now we're gonna talk about the different shapes of bacteria. All right, so there are 3 basic shapes of bacteria. However, we're gonna do some regular shape things, but 3 basic shapes. That's the takeaway for today. All right, now we already talked about the fact that bacteria are small, right, compared to eukaryotic cells. And so the average size of a bacterial cell is about 0.2 to 2 micrometers in diameter by 2 to 8 micrometers in length, right? And so when talking about the bacteria, they're going to be mono. Mono means one. Morph refers to the shape of something. So one shape, single shape, monomorphic. And then some can be pleo. Pleo refers to many pleomorphic, many shapes. All right, but for the most part, we focus on, in our lab work, with bacteria that have one shape, for the most part. All right, some can be a little different, and we'll talk about it when they are. Okay? So what are the 3 main types of bacteria? So the 3 main types of bacteria are the first 3, the bacillus shape, coccus, and spiral. Those are the 3 main bacterial shapes. So bacillus is the rod shape. Looks like Tylenol capsules. That's how I always remember it. Looks like a Tylenol to me. All right, and then pocket shaped, spherical, look like peas, right? Look like peas. And then spiral. So spiral is interesting in that it has varieties of a spiral shape. All right. So Vibrio, you're like, I heard that name before. Vibrio is like a bacterial name. Okay, so we're gonna talk about that. So Vibrio is a shape, looks like a comma, like it literally looks like a comma. The spirillum shape is like, it's a spiral, but it's loose. It's like a loose shape. It's like wavy hair, right? See somebody with some wavy curly hair? That's what it looks like. And then a spirochete is like if you have a screw and the ridges around that screw, they're like firm, they're rigid, but they're spiraling around that screw. You guys know what I'm talking about? Like you look at screws, right? Like I know I'm not the only one that screws stuff together. So with that, you know, spirochetes are spiral in their shape. They're more rigid, they're more firm, right? So both of these are spiral. The spirillum and the spirochetes are spiral. It's just that the spirillum is more loose, flowy, but it's still spirally, like curly hair.
So would spirillum look more like a wave, and
Yes.
spirochete would be more like a curl or a
Corkscrew.
coil?
Yes,
Okay.
like a coil, corkscrew. Yes. Yes, looks like wavy. Yeah. So those are the three basic shapes. Okay, so something this is gonna pop back out later, but I'll tell you right now since we're talking about it. So the name Bacillus, that sounds like a bacterial name, right? Bacillus anthracis. Okay, so that's gonna be a thing. You're gonna see some bacteria that have the genus name may be the same as the shape, and that's fine. Just making sure that when we do, this is gonna be important for writing stuff out. So like on our exams, if I have you write this out, or even in lab, I have you write this out. If you're talking about the bacillus shape, that can be lowercase, right? If you're writing in a sentence, it could be lowercase. It could be capitalized if it starts off the sentence, right? But you don't have to worry about italics and underlines because you're just talking about a shape. Same thing with Vibrio. Vibrio cholera is cholera. The infection, cholera, the contaminated water condition. Vibrio though is a shape. So if you're just writing about the shape of Vibrio, you can write that lowercase, right? And you can write it capitalized if it starts off a sentence, but you don't have to if it's in the sentence. Does that make sense? And I make that clear because people get that wrong. And I want to make sure you guys understand why it's wrong so that you don't do it. And I'm going to say it again and again as I need to say it. All right, to make sure you really get that. All right, so you do have star-shaped bacteria, but like that's not really common, so we don't really focus on that. And then you have rectangular-shaped bacteria too. We'll look at them, but that's not really common, but they exist, and we'll acknowledge them. Okay, so with the Vibrio, that's what it looks like. It looks like a comma. It's cute. All right, well, not the bacteria, but it's cute in theory. So Vibrio is going to be a comma shape. The spirillum is kind of wavy, and then the spirochete is more of a tighter type structure for this particular bacteria. So there's the star-shaped bacteria. Here's the rectangular-shaped bacteria. All right, so this is what I wanted to get to right here. Okay, so we're gonna talk about these. Make sure you guys know these. I swear, you're gonna have to identify these too. So, diplo. Diplo means two. So, diplococcus means I have two spherical shaped bacteria, right? Diplobacilli means I have two bacilli shaped bacteria, bacilli rod. So I'm going to say bacilli and rod is the same thing. I use them interchangeably. So bacilli, rod, same thing. So rod shaped bacteria. All right, so some bacteria can cluster together. Whenever you see the clusters, that's going to be staphylo. So that staphylo prefix is going to refer to clusters. And when I say cluster, it looks like grapes. Like some grapes that's just like hanging out. So staphylococcus is going to be clusters of coccus-shaped bacteria, and we'll look at that on the next slide. And then you have chains of bacteria, right? So you have streptococcus. So strepto would be the arrangement. That's the actual term, strepto. It looks like Mardi Gras beads, like one Mardi Gras bead to the next to the next. It really does. And so streptococcus, a chain of circular-shaped bacteria. Streptobacilli, a chain of rod-shaped bacteria, right? One next to the other. Tetrads, that should be easy. Tetrad, you play Tetris, don't you? Yeah, Tetris is 4 blocks every single time, right? So tetrads are going to be 4 groups of 4 bacteria. All right? And then you have cubes of bacteria, group of 8, and it's called Sarcina. It looks like a real cube. Like it might be cube, might not look good, but you know. Let's see what happens. See? Yeah, don't even do it right. So back in school, like, okay, I'm trying to think, in school we did like this is our cube, how would I do that in real time? That is a miss, but that's 8. I'm trying to do it like that, I tried. So let's look at the real arrangement for it, okay? The real one looks like this. Okay, so you have the diplo, and so when looking at it, it's showing the plane of division, right? So you can put a plane of division in between the two pairs. Well, the diplo, the pair of bacteria to separate, differentiate between two. You can do the same thing to the streptococcus chain. You can see you can put a separator in there, plane division, to see how that looks for a top view, a side view, just depends on where you're cutting it, right? And then you have your tetrads, the 4, just 1, 2, 3, 4. The cube, right? The sarcina, the 1, 2, 3, 4, 5, 6, 7, 8. You have two planes of division with that, right? Because of how you can cut that straight up and down, you can cut it sideways too, because you have them arranged in that order. And then you don't really have any planes for Staphylococcus. It's kind of just irregular in how they cluster together. Can't really do anything with that. And then we have this is for cocci. So the arrangement for bacilli or rod-shaped bacteria are going to be here, the single bacillus, diplobacilli, streptobacilli, as you guys see right there, that's chained up together. And this is one we didn't mention. Okay, so this is really not that common. So this is where it looks like a rod, but the ends are like really, really round, like a sphere. Right, so they are shortened though. They're not as long as the regular rod-shaped bacteria would be. So it's a little bit shortened and the edges are a little bit rounder because it wants to be a rod, but it looks like a sphere and it's like combined. All right.
So when the names are one word, then we do underline or italicize it?
Oh, none of this. No. So with these are just referring to the arrangement. So these are arrangements. So for instance, when you look at bacillus, right? So when looking at bacillus, the scientific name, right, it's going to be capitalized if you're talking about it. But remember, we only underline and italicize when you have the genus and species name together. We need the whole thing. So we need the genus and the specific epithet. That's the real thing. So we need the genus and the specific epithet together to underline it or to italicize it if we're typing it, right? But if you just see regular old bacillus hanging out in context, you capitalize it because it is, you know, it's a genus name, but you don't have to underline or italicize because there's no specific epithet following. Make sense?
Yes.
So then that's the shape bacillus, right? So I knew the slide was coming, but like I was already there, so I was like, let's just talk about it. All right, so bacillus is one that we'll see, and there's a picture of bacillus on the next slide. Bacillus anthracis. All right, so gram stain, bacillus anthracis. What color is that? Purple. So that means it's a gram what? Positive. Great job. So bacillus anthracis is a gram positive bacterium. Yep. All right, so let's talk about the structure of prokaryotic cell. We're going to actually go through all these structures, but see those little long appendages that extend from the body of the cell? Those are flagella, right? Notice how those flagella actually have some structures attached to it, embedded within the membrane. That's going to be something we're going to talk about in detail in just a moment. And so also when looking at the bacterial cells, you see those colors right there? We have these like black and we have these red colors. And so these black and red colors, the black is, well, let's do red first. So the red tells us that these structures are present in all prokaryotic cells. All right, the red tells us that they're present in all prokaryotic cells. The black tells us that they might be there, like, just depends on what you're talking about. Okay, so red is everything that's found in prokaryotic cells. Black is gonna be, maybe. All right, maybe, it should be, it might be. So in red, we have the cytoplasm, yeah. Ribosomes, specifically the 70S ribosomes, we'll talk about those. The plasma membrane, the nucleoid containing region, the plasma, well, I see the plasma membrane again. So yeah, those will be the structures that will be in all prokaryotic cells. Now, when it comes to cell structures that might be there in black, we have the pilus, we'll talk about that, the capsule, the cell wall, the flagella, the fimbriae, plasmid, we have inclusions, and yeah. Questions? So remember, in black, maybe. In red, it's there. It's there. All right? So we're going to talk about the structure structure and function first of the glycogen. I'm sorry.
Sorry, could
No,
you
no, you
repeat one more time for me? It was like cell wall inclusion.
That maybe be there, that may be there, sure. So the structures that may be there are the flagella, the cell wall, the capsule, the pilus, P-I-L-U-S is up at the top, we're gonna talk about that soon. P-I-L-U-S, that's the singular form of it too. Fimbriae, plasmids, inclusions, and then we got the capsule already.
Okay,
Yep, capsule and cell wall, you got those?
yes.
Alright,
Okay, thank you.
you're welcome. Alright, so with that being said, I'm gonna describe the structure and the function of the glycocalyx, which is gonna be fun. And then we're gonna talk about these different structures involved with movement and exchange of genetic material. Alright, so the flagella, axial filaments, and the fimbriae and pilus. Alright, so the glycocalyx. So the glycocalyx is interesting because we kind of talk capsules, kind of having that gelatinous covering. So it's really officially in the category of a glycocalyx. Alright, so the glycocalyx is a structure that is external to the cell wall, right? You saw that on the last slide, right? How you got the cell wall and the glycocalyx is there, if it's there. And so it's viscous and gelatinous. I just said that, the gelatinous covering for the capsule. Now, glyco tells us that it's gonna be something with a sugar, right? So it's made up of polysaccharides and or polypeptides. What's a polypeptide? Many proteins. Yep, lots of proteins, okay? So we know for a fact it's gonna be made up of a polysaccharide. So it's gonna be made up of a lot of sugars, or it may have polypeptides too. So it could be just straight sugars, or it could be sugars and polypeptides. So there are two types. So you can have the rigid, neatly organized, firmly attached capsule that we've been talking about so far. All right. And we could also have this other that's more, it's loosely arranged, not as organized as the slime layer that wraps around the cell wall as well, right? So it's still a glycocalyx, it's a different texture compared, and different organization compared to the capsule that we focus on, all right? So this is actually a very important structure, all right? Because this structure, if it's present, can be the determining factor as to how harmful a bacterial infection can be. So, what's the function of a glycocalyx? It actually contributes to the virulence. The virulence tells us how deadly or how dangerous or how infectious a particular pathogen can be. All right, so the virulence can tell us how infectious that particular pathogen can be. So, capsules prevent phagocytosis. I know that's like a way back word, but like, do you remember that word, phagocytosis? What do you remember about that word? Cell eating, yes, great job. So cell eating, right? So capsules prevent the cell from getting destroyed, right? Prevent the cell from being eaten or destroyed by a type of a immune cell, right? And so this also is great for the microbe because it can attach to different surfaces, right? It can attach to different surfaces within the body. And so what are the bacteria that have a capsule present? So Bacillus anthracis has a capsule, Streptococcus pneumonia has a capsule, and Klebsiella pneumoniae has a capsule. So one thing that's really interesting about Streptococcus pneumoniae, it can actually exist without a capsule, and it won't be virulent. It's okay, if that makes sense. But once that capsule is on it, oh, it's a problem. Because you can treat it without the capsule. Without the capsule, you can treat it because you can destroy it, right? It's gonna be easier to treat it, and that's why I tried. All right, because it's more difficult to treat that one. You treat it, just more difficult. You treat it. So, question?
So the calyx part, was that talk about its shape or like what it does?
So the
Is it like a
calyx-
horn shape?
So the suffix calyx,
Yeah.
let's double check. Because I don't want to lie to you. I know glyco refers to the sugar. Calyx, when looking at it, let's see what this bad boy. Put it in my dictionary. Okay, so it's basically referring to the outer covering in my bacteria. Okay, so the calyx is the husk. All right, so it's basically referring to the structure itself. So the calyx refers to the husk, and the glyco refers to the sugar. So it's a sugar husk. Yeah, great question. All right, so you have the EPS. The extracellular polymeric substance, I won't call it EPS, but that's what it's really called. I won't do it on the test though. So the extracellular polymeric substance, it helps form the biofilms, and we see that within the glycocalyx. All right, so the glycocalyx helps with the formation of biofilms. So if you remember, biofilms allow things to be trapped in certain places, right? So a biofilm layer can exist, and underneath that layer could be different types of bacteria, and these bacteria can be pathogenic bacteria. And so when thinking about a biofilm, biofilms will protect the cell, helps the microbes attach to surfaces. So for instance, Streptococcus mutans. So Streptococcus mutans is the causative agent for dental cavities or caries, right? So for instance, you guys remember going to the dentist when you were little and you would brush your teeth and they would give you the little blue tablet and you would chew it and your teeth would be blue or purple or red? Did y'all do that growing up? Okay, that's normal. Okay, so when you did that, you see like that little slime layer on your teeth, and that kind of highlights to the doctor like what's going on if you have some extra plaque buildup, some things with your teeth, to where, and it also can help show whether or not you have these like different like, I'm not gonna say irregularities, but like kinda. So that way, once they saw that slime layer, they washed it off and they got all that filth and not filth, all the grime and the plaque off of your teeth. All right, so the biofilm layers are actually there to protect the microbes, but if you remove it, you get those microbes off, right? And so Streptococcus mutans is the causative agent for dental cavities, and then Vibrio cholera, like I mentioned before, can cause Vibrio. And so these guys, that's why it's a problem with Vibrio, it's water, right? So biofilms in water, that's great for the microbes, bad for the people that consume or interact with that water. All right?
So they're important because they protect the microbes?
Absolutely, they're important because they protect the microbes and allow the microbes to continue thriving in that environment. So biofilms can also form on pipes, right? So at our homes, if you look down your drains, it looks slimy, you might want to clean it because that will allow for bacteria to be trapped underneath there. Or not, pour something on it. Like, you don't have to like go in there and clean it because you don't pull the bacteria out. Just pour something in there. All right, so I'm sorry.
So is it like basically if someone wore armor and then they put like lubricant on the armor and they're like try to touch me now? Is that kind of the vibe?
I mean, okay, so if they do that
Yeah.
and they have lubricant on the armor, can people still like penetrate the lubricant to get to the armor? Is what you're like?
No, because it's a hard armor undershell. So
Uh-huh.
it's like if armor was also
Because you're already,
slippery.
yeah. I get that. That's actually funny.
So they.
Essentially like that. The only thing would be with if I were talking about that analogy,
Yeah.
I would say the biofilm would be the armor itself,
Okay.
and you as the person wearing the armor would be the microbe that's protected,
Yes.
right? And so the slime on top of it would be like an antimicrobial or something cleaning. Like that's not gonna do anything because you got the armor on.
Okay.
Does that make sense?
Yeah.
Because I'm like, that's how I'm interpreting from how you're asking me. That's odd that. Okay, good deal. Any other questions? No? All right. You guys are getting tired. We're gonna stretch in a second. All right, so when looking at this picture here, the micrograph, remember I was saying Streptococcus pneumoniae, if the capsule is present. So here you see the capsule present within that particular structure, and if it's there, this is a very, pathogenic strain that's very difficult to treat. All right, you can treat it, definitely can treat it, but it's more difficult to treat because of that capsule. All right. So let's talk about our flagella. So we've mentioned the flagella before, right? But now I'm gonna go into some details to its actual structure. So I do want you guys to pay attention to that. That is on your study guide. I asked some detailed questions about it. So the flagellum is filamentous. It is an appendage that is on the cell surface. It looks like it's on the cell surface, but in actuality, it's embedded within the membrane. So it's going to be able to move the bacteria forward in a whip-like motion. All right, in a whip-like motion. It's made up of a protein, flagellin. All right, so it's made up of a protein, flagellin. Flagellin is not the right name, flagellin. So when talking about the parts of the flagella, remember these three things. We're going to look at this on the next slide too. So of course you have the part that we see. All right, we have the part that we see, which is the filament. And then you're going to have the hook portion of it that's attached to the filament. But this is what's embedded within that particular cell wall and membrane. So the basal body is going to be anchoring the flagellum to the structure, which is why it just doesn't fall off, right? It's not connected on the outside, it's actually embedded into the cell wall and membrane. So it has different rods or pairs of rods, rings, and then different rods within the flagellum. It depends upon what we're talking about. But it has rods and rings on that particular structure. And so the reason why I make it a notion to say, like, depending upon what we talk about, because we have two main categories for bacteria, right? We have gram-positive and we have gram-negative. So let's look at what this looks like for our gram-negative bacteria. So for our gram-negative bacteria, we see that, and then it's gonna be different because our gram-negative bacteria has two membranes, right? Remember, it has its regular old plasma membrane, it has its peptidoglycan, then it has another membrane outside of that. Remember I told you guys that? So because of that, it's gonna be a different arrangement within the membranes for this particular flagellum structure. So you have your flagellum attached. So you see your filament, that's the part that we see when we look at it under the microscope. You have the little hook portion that connects the filament to that structure for in which the basal body will be able to embed within the membrane. And so looking at this, you're gonna have your different rods and ring structures present. So the basal body is embedded within the outer membrane. You have a portion of it on the peptidoglycan layer, and then you have it within your inner membrane. All right, so this is all your cell wall. So your cell wall is the composition of the outer membrane, the peptidoglycan. Right, so cell wall, these two. Plasma membrane hanging out right here, and your cytoplasm, the fluid on the inside of your cell. All right, so what ends up happening, the reason that your flagellum moves is because this rotates. That rotates. And when that rotates, it allows the flagellum to move. All right, because that rotates. All right, so when looking at this, you notice that your gram-negative bacteria have two pairs of rings. So we have two pairs of rings right there. 1, 2, right? Two pairs of rings. And so gram-positive, how many think they will have? Maybe less, right? Because it's only one membrane, right? So when looking at your gram-positive bacteria, this one is going to have, of course, your filament, what we see, the hook that allows for our basal body to go ahead and connect and embed into that membrane, but it's only one ring. And that one pair of rings. And this pair of rings hang out within that plasma membrane. All right, it hangs out within that plasma membrane, does not penetrate it does not anchor in the cell wall. The cell wall just consists of your peptidoglycan, the very thick peptidoglycan layer. And with the basal body, you see that the hook is gonna rotate, flagellum, move. All right, so that looks different, right, between gram-positive and gram-negative, and that's because the gram-negative and gram-positive cell wall structure is very different. Let you guys get some air, take a break right quick for a moment, stretch, and I see we're getting eyes low, and we're getting hot, so stand and stretch, get some air to make you happy.